Hello, good morning, everyone. Those of you who are on the West Coast, and then good afternoon, almost good morning also on the, like I think it's a little bit early for those who are on the East Coast as well, and good afternoon for those who are in the other parts of the world. It's welcome to our e-seminar series on Translational Biomedical Engineering. It's our great pleasure to host Professor Mina Hoorfar from University of Victoria. Today at these e-seminar series, she will be talking about breath sensing for personal health monitoring. It's a very interesting topic that I also have personal interest in it. So I'm looking forward to your talk, Professor Hoorfar. Before we start, and as usual, if you have any questions during the presentation, please use the Ask a Question box. It's right at the bottom of your screen. Also, you can share your thoughts and contact chat with our speaker and us using the chat box, which is on the right-hand side of your screen. Next week we have Professor Sanjay Raj from the Department of Mechanical Engineering and Bioengineering at the New York University, Abu Dhabi, with us. So we're looking forward to his talk. Make sure that you follow us on Twitter with our handle TransLBME. You can also follow me and Human on Twitter and LinkedIn to get most up-to-date information about these series and the upcoming speakers. Last but not least, I'd like to thank our sponsor, Montreal Transmedtech Institute, who has been with us since almost the beginning of this series, which was June of 2020, almost 2 years ago. So this is our 18th talk, and then we have a list of other speakers coming up pretty soon. So thank you, Montreal Transmedtech. Excuse me. So with that, I'd like to introduce our speaker, Professor Hoorfar, who is a professor and the dean at the Faculty of Engineering and Computer Science at the University of Victoria. In her lab, she leads a microfluidics and nanotechnology laboratory called MiNa, which is I think it's a coincidence that the lab name is the same as our speaker's name. So she's known nationally and internationally for her research and innovation in the areas of flow for her research and innovation in flow in microstructures and flow and heat transfer, mass transfer in microstructure, straddling the fields of microfluidics, biochemistry, and fabrication of biosensors, and with specific focus on gas sensors. Her multidisciplinary research has resulted in wide and varied applications, such as easier and faster detection of pathogens in drinking water and air quality monitoring in collaboration with industrial partners in oil and gas, municipalities, and life sciences sector. Professor Hoorfar completed her PhD at University of Toronto. She did also her master's at the same university, but before that, she did her undergrad at the Department of Mechanical Engineering in the University of Tehran, which in Iran we call it the Harvard of Iran. Well, there's always a competition between Sharif and University of Tehran, but well, I mean, this is I think a pretty impressive and high quality university. So with that, I'd like to first share my screen. Let me see, and then I would like to give the virtual floor to Professor Hoorfar.
Thank you so much, Dr. Mohseni and Dr. Salwaji, for inviting me and this amazing platform for sharing our ideas. And I call it dream. I'm sharing my dreams here, actually. And I have many of those dreams at night regarding the personal health monitoring. As Dr. Akbari mentioned, my name is Mina. And there are names here at the bottom. Currently, right now, I'm at the University of Victoria. I started this position as a dean July 2021, which I'm very honored. And my lab, as he mentioned, is microfluidic nanotechnology, and short for MiNa. Of course, we really try to find a name that matches my first name. It was coincidence, but also a little bit effort there. Before I start, I wanted to apologize. I couldn't share my screen, so Dr. Akbari I'm going to trouble you. So every time I say next slide, then you just press it. So let's go to the next slide. I'm trying to give a very holistic view of what is out there in terms of breath analysis, but also from the personal health monitoring point of view. The literature now is saturated, or not saturated, is rich enough, and there are lots of areas we can work on. So I think this is the place that now is the time to have a step back, have a good view of what is out there, what are the challenges and opportunities that we can target. So I start with, talking about breath sensing. To start with, actually, what is in the breath, that what we're trying to sense. What are the methods that we can use for the breath sensing, the most common one? And then after that, I am targeting three diseases: diabetes, lung cancer, and COVID-19. Of course, the latter one is because of this recent pandemic that we had. Also, but there are another reason is that the diversity of these diseases. Diversity from the point of view that the mechanism of these diseases are different, from the metabolic aspect to the cancer, which is very different than diabetes, to the viral infection, similar to COVID. And I wanted to bring these three different diverse diseases to show that how breath analysis or the detection of the biomarkers in the breath can or may not or cannot be useful. And at the end, I would like to just put a few slides, my dreams or the proposed solutions. Of course, by all means, they're not the maybe the most viable ones or the most doable ones, but feasible ones, but the ones that what I mean by I and my team members are thinking about it, and we're trying to push the boundary. So next slide. Breath is a very complex mixture. We always thought that the breath has mostly nitrogen, CO2, and oxygen, of course, but is way more complicated than that. Each breath has thousands of volatile organic compounds, certain mixture of inorganic gases, non-volatile gases, and humidity, of course. Exhale breath has a lot of material in it. So next slide, sorry. If we wanted to look at it, here is a good summary of what, in terms of the categories of the molecule we have in the breath. On the left-hand side, you will see we have saturated hydrocarbons like ethane, pentane, basically carbons and hydrogens. And the second column is example of the production mechanism of these in the breath. And then we have unsaturated hydrocarbons like isoprene, which you will see that in some of the areas they're very useful to measure the amount of or the biomarker for the diabetes. Oxygen containing acetone, the same thing. People are using it for the measurement of an indication of diabetes. And for example, acetone is because of the lipolysis or lipid preoxidation. You've heard about ketosis. These days, the intermittent fasting and all of these diets, the area of the ketosis coming out. And this is one of the biomarkers that you can see. Because of the ketosis, you will have it in the breath. And then we even have the sulfur containing, the first time I've heard it, I was really shocked that we have sulfur containing compounds in the breath, like ethyl mercaptan. And ethyl mercaptan, you use it in odorizing natural gas. Of course, not at the same amount we have in the breath, but there are some amount of breath because of certain incomplete metabolism of certain diseases can happen in the breath. And finally, nitrogen-based ammonia, and again, due to the certain diseases. So we have a sort of category of different molecules, and in each category, you have different gases, which can be or may be related to certain mechanism in terms of their production. Next slide. I really want you to pay attention to this slide because I will use the name cluster a lot during the presentation. Here I have a few diseases, and we listed 21 common volatile organic compounds in breath that you will see in many diseases, and they are a little bit more than other VOCs and other gases. If you look at this diagram that my group actually, they published it in their review paper, certain biomarkers or certain VOCs are for diabetes. And then for COVID, we have something for heart failure, lung cancer, cystic fibrosis. So what we're trying to say, there's a cluster of biomarkers that you, or VOCs, that there are intersection between different diseases. You cannot pinpoint one VOC and say, oh, if I detect ethanol, then definitely it is related to lung cancer. Or if I detect acetone, and I will get back to that, that will be definitely diabetes. It's a cluster. And all of these gases, they may, or VOC, they may be in other diseases as well. So I get back to this notion of cluster or cluster finding of the gases later on, but it's very important to remember this diagram when we move on to the diseases that we're going to talk about. Next slide. So now we know what is almost in a 30,000 feet view, what is in the breath. So how we can measure it. Of course, whenever you wanted to measure in terms of biosensing, it doesn't have to be breath, but any body fluid, you need, of course, the analyte, which could be the breath, could be blood, could be saliva. That is your analyte that you're trying to measure, for example, the acetone in the breath. And then you need a recognition element. That's why it's called biosensing. It's not just the sensing. A recognition element, that this recognition element, as you see in this right-hand side in the middle, can be enzymes, could be antibodies, could be nucleic acids, could be even the whole cell. So what it happens is like a lock and key mechanisms. Your analyte fits into your receptors or the biocomponent, and then from there, they create certain signals. And they happen at the sensing material. This sensing material could be metallic, could be carbon, nanocarbon tubes, could be anything that you can, or it could be RGO, the reduced graphene. But there's a sensing material that all of these two analytes and the receptors, they come together and then they create a signal. And as a result, you need to decipher this signal. So you need a transduction, you need a transducer principle. Whatever signal they're producing, you can make it as a way to read out. So there are many different transducer principles. You can use optical, mass-based, colorimetric-based, magnetic, electrochemical, acoustic, something that eventually you get a signal out of these interaction. And then, of course, we can use the AI, machine learning, to classify the signal and understand what happened from the beginning. So that is a big mechanism of any personal health monitoring. There could be in some of them one element missing. For example, some of these monitors, they don't have the bio element, so there's no recognition element. Just the analyte comes at the sensing layer. The interaction happens, and you have a transducer, and then you have a readout. In some of the cases, the readout is just yes or nay, so there's no machine learning. But this is a very overall picture of what we're talking about, personal health monitoring. Next slide. The research on breath monitoring has actually increased over the years since 2006. So before that, breath as a body fluid was not very attractive to be used because there are other body fluids like blood, saliva, urine. Blood, of course, is invasive, but urine and saliva, they could be gathered in a non-invasive way. But the importance of the breath in terms of what it carries was not recognized till 2006. Or maybe we didn't have sensors that could measure anything we have in the breath with some accuracy. But anyway, regardless of the reason, it's from the 2006, the number of the publications sort of increasing and coming up. And especially last 2 years, because of the COVID, and it is a respiratory disease, you will see that loss of interest from the research community to come up with some sensors for the even clinical setting or the personal monitoring to use the breath as a way to measure what is happening in the human body. Next slide. So I said, as I mentioned, this is an overview of the breath, what entails, what can give us. So I'm going to dive into the 3 diseases I told you. We start with diabetes. Is a metabolic disease because it's related to the insulin, whether insulin production or lack of production or insulin regulation through the beta cell in your pancreas. It's one of the most expensive in terms of the pharma kind of disease in any society, and the number of the people with diabetes is sort of increasing every day. And there are 2 types of diabetes. There are type 1, as you may know, and type 2. The difference is that in the type 1, your beta cells, they are damaged and impaired, and they cannot even produce insulin. So there is no production of the insulin is basically insulin level decreases. And then the type 2, which is there's a resistance towards the insulin. So the regulation of the insulin is not done properly. So at the end of the day, it doesn't matter. For us, that we're looking at a way to measure or come up with an indicator to correlate this mechanism of insulin problem in the body, I call it, by breath. So next slide. But before I get to the breath, I wanted to really show this this table, which we actually spent a lot of time to gather this table. This is also published in our review paper. So if we put aside the breath analysis, let's see what is available in order to diagnose diabetes. Is a disease that's been diagnosed for years and years and decades, so it's not something new. The method that they're using without breath analysis is invasive glucose monitoring, and glucose directly will tell from the blood that, or from the skin, if you measure the glucose, that what is the function of the insulin and beta cells. And there are different methods here, and they're all compared to the gold standard way of measurement. And you see that the second column from the right shows the performance of each of these methods. And we use a sort of parameter called MARD, or the mean absolute relative difference. The smaller the MARD, the better, means that you're closest to the gold standard, your method. Of course, this, we cannot really come up with a conclusive sort of idea which method is better than the other one, because in each study, different groups have done it. The sample collection might have been different. The number of the participants are different. And as you know, anything about clinical tests or anything involves a human, depending on what is your sample pool, what range and diversity of the sample pool you're dealing with, you might get a different sort of performance. So I know it's sort of a table with some of the value and percentages, but what I'm trying to say is that a lot of still a study required for invasive way of measuring or monitoring diabetes. Next slide. Now, if we add another complexity to it, I wanted to say, let's forget about the invasive way of measuring the diabetes or diagnosing diabetes, and we go to breath. Of course, we cannot measure glucose from the breath. And we know glucose is the one that you want to measure to know that how is the status of the diabetes or insulin production or lack of production. But what people have done, they found over several studies that when there is diabetes or there is some kind of indication of impairment in the pancreas, there are certain biomarkers will be present in the breath. Of course, depending on the type 1 or type 2, some of these biomarkers may vary. But among them, acetone seems to be very present. And that's why you will see in the market in the past decade, there are breath analysis for diabetes. But I'm going to use the word, and please remember this, a standalone way of diagnosis. What it means that they measure only one VOCs, which is acetone. And from there, they say the person is type 1 or type 2, or whether they have the diabetes or not. So this standalone way of measurement and diagnosing diabetes could be dangerous, because there is, as I mentioned, that slide with the circles. There are, as I say, intersection between different diseases. One biomarker may not really indicate what is happening there. And again, remember, to begin with, acetone was not the real indicative of the diabetes. We had glucose. So going with one biomarker could be very dangerous. Next slide. In fact, people have shown that acetone is the byproduct of what's happening in the pancreas, as it may not be really related to diabetes. It's a byproduct of the blood ketone. Do you remember when I was talking about intermittent fasting or keto diet, that you've heard about those? When you go to the ketosis, means your body starts burning fat, then you release acetone. And that may not be exactly the right correlation with diabetes and with the insulin in the body. And there is actually right on the top, there's a figure that shows the correlation between the acetone in the breath and what we are trying to do in the insulin way is 0.68, less than 0.7. And that's not a good sort of correlation. So that's why it would be very dangerous if we just use acetone as a way to say somebody has or is diabetic or not. Next slide. So why Because, as I said, the factors influencing the cell body, if you look at the right-hand side diagram, the circles, there are many factors: age, physical activities, diet, muscle mass, gender, ethnicity, whether the person has a smoking behavior or other co-metabolized diseases, air pollution even. So it's not just one that we can say, let's target all of the group of our participant who are in different age group. No, age is one of the factors. Physical activity is another one. And I'm sure that there are other factors that can affect the amount of acetone, and as a result, skew the acetone detection as a diabetes recognition in our body. And you will see that we have on the left-hand side a table we summarize in our paper that shows these BRAs, or brace we call it. The mean brace is the breath acetone concentration, ppm, and how different techniques in body, or people with different diets or fasting or caffeine or tea drinking or after sleep or before sleep or during exercise, how much this breath acetone concentration changes. And they are not the ones that might have diabetes even. So that's why I think it's very important to have a better come out of this and have a better view of what we're trying to measure and what kind of these devices we need to design. Next slide. So that's why when we saw this, we said, okay, forget about standalone VOC measurement, which was the acetone so far. Let's look at other research areas that somebody did a bunch of or cluster of the VOCs. And there are actually areas or work or studies that we found people are doing it. For example, this is the table we summarize in our paper showing that the cluster, the left-hand side column shows the biomarker clusters. You will see that's not just acetone. There are different ones. Of course, acetone presents in many of them, but you have isoprene, you have propane, and also the different types of subjects, 17 healthy or 130 healthy and 70, depending on what funding they had in terms of hiring the subjects, and then what methods they use. These methods, all of them are bulky methods like gas chromatography, and some of them are the analog semiconductor sensors. And they found that certain correlation between what they have measured and what the diabetes is. So this is a very good research to show that, yes, there is a possibility to use breath for finding the relationship between the insulin level or the type of diabetes or your diabetes, as well as what is in the breath. So this was very promising. However, we still need to do more work. So next slide. So what we have in terms of challenges right now, if you wanted to look at it and see that where are the opportunities, it's that in all of these research areas, even the previous slides, the table that we have, we only have 4 studies. The population level survey is very important. And unfortunately, different groups are doing different ways, and they select the data or they collect the data differently. They pick their participant differently. So you cannot really come up with a conclusive remarks that whether this set of cluster of biomarkers are going to be an indicative of the diabetes. So that's why we need to have a very thorough design of experiment in terms of population level survey and see that what is the minimum population we need, and what are the factors in that circle that I showed you are the most important one in the diabetes detection. And then create a biomarker database based on that, which we are lacking right now. And sampling method is very important. If you get the sample of the breath and store it, and tomorrow do the analysis will be different than you do it fresh. If you have humidity, which is another one interfere with your measurement, that could be different. If you use a certain plastic as your container, you may lose some of your VOCs. So sample collection and sampling, it has to be standardized, which we don't see in all of the study that we are gathering. And again, the volume of the sample and what is the pushing the limit of our detection. Right now, the limits of the detection, the sensor we have may not be enough to really see the effect of certain biomarkers. And of course, for all of those, we really need good pattern recognition systems to really analyze the signal that we're getting. So these are the challenges and lots of opportunity for people with expertise to get into this market and make the better next generation of breath analysis or breath analyzer for diabetes. Next slide. Now I'm shifting to totally different disease, lung cancer. Um, lung cancer, as you see on the top, there are of course the stages, and we know that depending on the stages, the level of the VOC and the type of biomarker even can change. And but the point is, the dream is if you can detect it in a non-invasive way as a way of personal monitoring early, then you can diagnose it early, then you can start the treatment early, and then you can improve the outcome and the survival rate. That is the whole goal. Of course there are invasive way to do it, but all my talk is about personal monitoring in a non-invasive way. So what if we can use the breath and figure out what are the VOC depending on the stages relevant to the lung cancer. So let's go to the next slide. Thank you. So again, we get back to that cluster. Remember cluster that we have, similar to diabetes, is not just one standalone VOC or biomarker that we can do lung cancer or LC. I sort of sometimes say for short. So there is a one type of research using the VOC, the breath. There's another type of research for the lung cancer that we use the breath condensate, or short for EBC. And that one actually gives you maybe more accurate result because you get the condensate from the breath, then you use this as a way to get the protein, RNA, DNA, a sort of profile of your cell from the condensate, and you come up with a sort of solution. Of course, it's more involved in terms of the gathering and analysis, but I wanted to point out that for the lung cancer, both research areas are advancing. We don't have such a thing like a condensate for diabetes, but I wanted to bring it up. It's not just the VOC. You can also target the EBC, but the EBC is more sort of complex than the VOC monitoring. Next slide. Now, this is a table that we found summarized out of many studies that what are the current sensors? So I'm just talking about the VOC measurement because that's my target and goal and dream of using the breath gas to measure, not the condensate, but it could be in future both of them together. So this is a table summarizing people who are using VOC associated with LC or lung cancer. There are different methods that you can see that people are doing. And again, they're using different VOC detection. It's not just one standalone. The subject numbers or population survey number changes, and then the accuracy depending on the two factors they're doing, sensitivity and specificity. Sensitivity means how low ppm you can go, and the specificity means that whether you're detecting lung cancer or other things in the body. So that is very important to do. So there are studies, and you will see that the research in this area is really promising and going forward a lot for the VOC associated with the LC or lung cancer. Next one. And also there are sensors right now in the market that you can see. There are colorimetric sensors. So if you remember at the beginning, I talked about the transducer part of the biosensor. There are some of them colorimetric, even with the color, they use it and they say, okay, you're at this, these areas of the colors will tell you that what is the stage and whether you have in your breath a certain biomarkers, VOCs, or there are some kind of electronic noses. Electronic noses are sort of series of thousands of sensors that they turn on and off, and based on the interaction between different signals, we get or certain pattern are recognized, and we get the results of what is the outcome in terms of the VOC associated with the LC. So there are also sensors available for this in the market. Next one. One of the two interesting ones that are right now in the market commercialized, we sort of came into or ran into, are these two sensors. Both of them, they're measuring the VOC, not the condensate, breath condensate. And the left-hand side is using the photon ionization as one of the methods to sort of get the signal of the breath and ionize it and get the molecules that they want in terms of the VOC and measure it. And the right-hand side is the one that is using the colorimetric one and the gas sensor array, that the breath goes to certain areas of the sensors and then they measure it. So these are actually already available in the market for the lung cancer. So you see that there's a huge market for the lung cancer associated VOC. Next one. But again, I want to go back to the challenges similar to diabetes. We don't know what is the group, not even standalone, a cluster of the biomarker associated with the lung cancer. We don't know that. So there is a lack of database for the lung cancer in terms of the VOC. And that comes back not just because there is not enough or there's no accurate or sensitive biosensor available. No, there hasn't been done a systematic analysis with the right survey population to gather this database. So a very good research in the next few years would be somebody dedicates to create the biomarker database for one of these diseases, such as the lung cancer. It will serve the biosensor community significantly. And then, of course, as I say, for the reliability and accuracy, we need a good population level study. Concentration has to go low. So for people who are in the sensor business, we need much more sensitive sensors, because for these kind of cancer, if you wanted to go earlier stages, definitely those biomarkers are at the ppb levels. So definitely we need the low concentration. And again, how you sample, get the breast sample and use it for your analysis. There must be standardized way of doing it. For example, I learned that, and I don't have it any slide here, when you wanted to measure the impairment due to the marijuana smoking and measuring the THC in the blood, there is a standard way to do it. Certain type of needles should be used. It has to be in a setting that is a special clinic. So the same thing we need to really publish to say that what are the standardized methods for these diseases to collect the breath to do that. Next slide. So now we're getting to COVID-19. I know I'm using this because of the disease is one of the hot ones these days, but I don't want to just look at it COVID-19. I'm trying to target all of the viral infections associated with the lung diseases. And before COVID, we actually had other diseases in mind, but now this came and we say, why not? Let's look at what it's there. But of course, next slide, it's very new. You don't see many biosensor-based technology available from the breath. Most of them that you see, actually I have at home, are these nanotechnology-based biosensors that, from the body fluid, whether it's saliva, whether it's from the nose they gather or from the blood, they have sensors that they are loaded with certain antibodies that they are specific to the proteins outside the COVID cell. And by the way, those proteins are not very specific, but they are. And then when you have the sample of the cell, the COVID virus there, then it comes and then attach, and then you get certain signal. There are different ones available, and you can see that here what is working. Next slide. There are also ones that they are based on, as I say, field effective. This is a very accurate, more accurate than the other one, because they use the field effective transducers or sensors. And again, they have the gate, they have the two electrodes, and they load it with a certain very sensitive sensing materials. Some of them are graphene based, and then they load them with your antibodies related to, again, the whole cell protein, and they measure it to do that. This is another one we found as a nanotechnology-based biosensors. Next one. The only one we found, so this is another one, again, nanotechnology. I'm just bringing all of the things available right now in the market or in the research areas or research labs for the COVID. They're using, for example, nanowires, field-effective transducer. In all of them, just the sensing element changes, but the protein, there's a receptors, and that receptor is basically measuring or connects to the protein on the surface of that one. And then we have the electrochemical sensor as well, which is there coated on the multiple carbon nanotubes. So the only breath analysis based one that we found, next one, is this one, which again is a biosensor. So the breath comes out and it goes to a sensing layer, which is coated with the antibodies associated with the cell wall protein. And then from there they measure. So what I'm trying to say, there is not just the COVID. If you go to the next slide, there is a huge area or sort of opportunity here to look at the viral infection and the VOC associated with it. You see that there are not much, not only in the market, but also in the research labs associated with this. And then we looked at what is the mechanism. Is nobody studied ever what happens when you have viral infection? In fact, people have studied, if you just press the button enter, there are many studies that not for the detection, but they have looked at what is the mechanism of if there is any VOC production during viral infection, and there is. And I have the picture of when the virus comes and trying to infect a cell, they release odors, VOCs. And if you can measure that VOC, you should be able to, detect the infection. Whether that VOC again is a standalone or is a cluster, that's another question. What are those VOC? So there must be a study done there, but there is hope here. As one of the dreams I have means that viruses, when they infect the cell, they releases VOC. And there are studies done for different viral infection. Next one. So with all of these, with which we have done actually recently, this holistic review of what is available in the literature, and I wish we would have done it maybe 20 years ago, but of course, maybe the research was not there yet. But now we have very good understanding that where we are standing in research, in commercialization. Then my group actually, based on whatever we've done so far, we've gone back to the whiteboard and said, okay, this is what available in the market. This is what available in the research lab. What can we do in MiNa Group in terms of breath analysis? And these are the rendering of the devices that we have we are producing right now. They're mostly phone-based because we wanted to make it easy and more personable for the signal. And you see that we're not targeting one standalone. For example, for diabetes, we're doing the bunch of different analytes, and then from there, to go and see that what it is. But in addition to the analysis of the whole cluster, we're also doing lots of advancement in the sensitivity and selectivity and the material of the biosensors. So what is inside the box, we do lots of research. In terms of the brain of the sensor, we do a lot of research in terms of data gathering, data mining, as well as the data analysis or analytics. And then from the signal producing and the look of it, we also have lots of potential advancement there. So next slide. I would like to sort of picture some of the things that we're doing. The technology that we originally came up with, I think it was 7 years ago, or maybe even longer than that, now it's been licensed by a company, Cannabix. They're the ones that they're making the THC-based breath analyzer based on our technology, and this is their device for the road safety monitoring of the impaired. And it's basically based on a microfluidic channel that it's coated with certain layers to create a sort of profile, a smell profile for what is the THC in the breath. And for that one, we have studied a lot of different nanotechnology and nano features along the channel, and to see how we can make it better and more selective or specific to the THC that we are doing. So that is the microfluidic detection that we have in our lab. Next one. Other areas that we're working, they are more from the, not just the breath, they could be their electrochemical that we're doing from the skin, or we are doing from the breath even. And there are wearable devices that we're designing for that. There are, of course, the GFET kind of biosensors, and we have samples here. These are our own actually devices. And in right-hand side, for example, from sweat, we are measuring cytokine. And that is a way to show that the body is stressed for some reason, lack of sleep, exercising, doesn't matter, or even viral infection, your body production of this biomarker increases. And then we have a device, you will see that down bottom, that we can measure it. This can be done as a wearable device. You will see that on the left-hand side, which we can have a flexible polymer with this sensing layer and the aptamers that we design for these particular biomarkers to this, and then people can wear it like a Fitbit and they can measure it, and it can be used for different applications, similar to the lung cancer. If you remember in one of my slides, I showed that from the skin, they have done the lung cancer biomarker measurement. Next one. Now, this is a new one, and I know it's a little bit science fiction, but I have recently, because of COVID, we're coming up with the gasochromatic basically kind of sensor. You're wearing the mask, and based on the breath coming out and the viral infection VOC that I mentioned, you can get it colored. Of course, as soon as it should be in a way that people don't see it, it's just you see it. But the idea is that if you have the right biomarkers and a cluster of biomarkers, and also we know that what VOC we're measuring, you can create even the colorimetric way based on these gases there. So that is another area we're working right now. Next one, which is the last one. We are also facing challenges with our own technology. As I mentioned before, humidity is one of the big challenges, and we were very successful. The right-hand side, you see, creating for all of our sampling devices a humidity regulatory membrane. So this membrane, the gas goes without removing your gas of interest, always regulates the outcome of that at a certain humidity. And as a result, all of your measurements are done regardless what is the humidity of your breath versus my breath, or where I'm located. The measurement are done all at the same level of humidity, which is very important. That will increase the accuracy of our measurements and reliability of our measurements. We are also looking at the biomarker clusters, not just the standalone ones. We're really pushing the sensitivity of our sensors by making the GFET ones or the field effective ones. The one of the areas that we really need to work on is the recovery time. If you remember from the cancer, lung cancer usually takes about 18 minutes to get the signal. In some areas, that's too long. And we're trying to also increase the response and recovery time of our sensor to make it dynamic. And again, selectivity or specificity and sensitivity are always the important aspects. We are also trying to incorporate both VOC measurement and breath condensate, because we can see from both of them, you get different information and you can fuse them. And we're really looking at the material side of the research and see that how we can, by modifying the microfluidic channel, improve our sensing and sensitivity. And then, of course, instead of just one sensor, arrays of sensor. So I stop here. I'm done. I sort of tried, hopefully, tell you what is out there and what we are doing, and open to questions. Thank you.
Hear me? No.
Yes.
Can you hear me? Okay, all right, that's great. Thank you very much for your exciting presentation, very inspiring. So I encourage the participants to ask questions from our speaker. This is a very interesting field, in my opinion, and then we have personal interest. I mean, in that, and then we're collaborating on some aspects of these projects with Professor Hoorfar. And I apologize. So I have a few questions that I'd like to ask from you, Professor Hoorfar. My first question is about I mean, you mentioned sensitivity is a challenge, and then you talked about humidity and the effect of temperature on the readings. But I have a question about the volatiles that are present in the air. And how will that affect the noise that is produced in these sensors? And then if you have done anything to overcome that challenge as well.
The short answer, yes, the impact. However, what we have in the atmosphere is way diluted than when you can, for example, if we just talk about the breath, what you sample. That's what I say, there must be a standardized way of collecting the sample. The way we do it in the lab, of course, there's a spit, actually, a tube that removes the spit from the breath, and everything goes to a collecting chamber, which is very well sealed. The one thing that actually help us is that what the gases, unless you're in a very concentrated area with certain gases in it, is so diluted that what you're concentrating in that volume is way more than the other ones. However, if you're not dealing with this collection, you're having, for example, open wound, and you're trying to do it, then that would be more difficult. Depending on what is the concentration of the VOC that your wound or the bacteria infecting your wound are going to release. If there are more than the outside sort of gases, then that's good. If not, then you have that interference with that one. One way to resolve that issue is that always look at your baseline before doing your measurement. So when I have a sensor, for example, in this room, before I expose it to the certain area that I wanted to measure, you have a baseline of all the gases in there. That is your reference point. After that, when you calibrate it, call it calibration, then anything you expose to it will be result of your area of interest.
Mm-hmm. Mm-hmm. Very interesting. I have another question about the reproducibility of the results that you get from your sensors. Can you comment on that aspect of your sensor?
Exactly. So that goes back to the ppm level.
Mm-hmm.
That goes back to the way that we collect the sample. But if you are really good, your student follow the protocol of always collecting the sample the same way. So that issue is away. But then if it's the ppm, with our sensors, we have challenges to go below 50 ppb, parts per billion. So whenever you go below 50 pp, the noises are so much, the reproducibility goes down. So you need to know what is the inherent level of the detection of your sensor. If you wanted to really push it below that, they won't be reproducible. For the rest of them, those that are gases above those, how we make sure there's a reproducibility is that our sensor works based on exposure and then recovery. For us, in order to make sure that is reproducible, we have to do a very good job in recovery. And how do we know the sensor is recovered? Because we go back to the baseline, the same baseline that I was talking about. That way, your sensor again is exposed, unless you pass the lifetime of sensor. And we have tested sensors actually for 6 months and still they're good to go. We haven't gone push more than that. That goes back to the lifetime of sensor. That's why any kind of gas monitoring device, not for biomedical application, for even oil and gas you buy, there's a bump gas. Every morning or every time you have to calibrate it, get the baseline, make sure that your sensor is good to go, and then make sure that you go back to the baseline every time after the measurement. So this way, definitely your sensor should be reproducible. However, some impacts like humidity, impacts like temperature, they can affect it. But what we have done in our lab, that membrane that I said will regulate the humidity. With our sensor, which is a semiconductor, is always at the 400-degree level, which may not be good for some application, but at least we know it's regulated temperature. So these there are tricks that you can do in your analysis and measurement and post-processing of the data. But for us, we have seen very good accuracy or reproducibility of our results. Yeah.
Okay, that's cool. So, and that's what they do with that breathalyzer that you mentioned, the company
They calibrated, yes.
Okay, so people can't smoke marijuana and drive anymore.
Apparently, I don't know. So I don't know what stage they are, but that device is real. So they made I'm so proud of them. They did amazing job.
Yeah. Because I was like hearing in the news that there's still no standard way of, because there are breathalyzers for alcohol, but for marijuana, that's something.
I wanted to make a comment about the alcohol one as well. So even with the alcohol one, you don't measure the impairment. What you measure, of course, is being standardized based on the blood over the years. You measure some indication of how much you have in your breath. If the police finds that you are impaired, then they have to take you somewhere and they have to do within certain window the blood test, because that's your gold standard for the court to stand. The same thing for the THC or marijuana breath analyzer for the road safety. When they figure out, before we had the breath analyzer, they were doing the sort of impairment test, walking and all of that. If they figure out, they take you to station and they take the blood test, and that will indicate. So we don't have to have a gold standard as a breath analyzer. We need to have something which doesn't give us the false positive a lot. That would be the problem.
I see. Okay, cool. So we have Professor Griffith from University of Montreal, who in the chat box, she thanks your talk and then thanks you for your great talk. But her question is mostly focused on your COVID project, and then she's wondering if you can use these technology to detect viruses that infect the eye, for example, or other.
Thank you so much, May. Very good. Actually, it didn't come to one of my dreams about the eye, because I'm all about breath. That's good, actually, you bring up, and I'm really open to collaboration. Do you remember I showed this slide that we have a sort of GFET on a polymer base, which is a flexible for the detection of the viruses. And we are right now not doing viruses. We're doing for the certain biomarker that the elevated cytokine. That can be done on the lens because those are very tiny on the lens. And they're transparent. So yes, we can put anything on any flexible. The point is that then somebody has to test them and see whether what is in the eye or in the tear or in the things that infect it, it really has the good indication of the COVID. Always, I think what I found, making the sensor is not easy. I'm not saying it's easy, but implementing them and testing them in vitro and in vivo, they are the ones that have to be done very carefully. But with the eye, yes, because there are many ways to do transparent polymeric sensors, and we can do biosensor to make it very effective as well. And right now we're doing with the sweat, so why not with the tear?
Fantastic. So, a contact lens.
Yes, May, please contact me. My email is my first name, Hoorfar @uvic.ca. I would love to have a chat with you. I love this type of, exciting, high risk, high reward based on our answered research.
So I just typed your
Thank
email in the chat box, mhoorfar@uvic.ca, and then I know May is an expert in eye diseases, and then she also was one of our speakers a long time ago. I think
Good.
we
Yes,
should re-invite her again and then see
absolutely.
what has been done recently in her lab. So we have a bunch of other questions from the. So the ice is being broken. I don't know how much time you have, Mina.
I have a meeting. I have to go for the hiring in 3 minutes. I'm so sorry. I couldn't.
3 minutes. So,
Yes, yes.
so if you don't mind, I'll ask Elham's question because she was first. So, what is your approach to overcome the debye length limitation in FET-based sensors?
Oh, that's a very tough one. The debye length for us is the coating that you can do on your GFET sensor is very important. You want to have that debye length as thin as possible for you. That can really help you with doing that. So that's why that sensing layer that you have in your between the two electrodes are very important. We are actually having one paper in the pipeline for the review that is exactly showing the effect of that debye length on the sensing. Of course, it was done not on the sweat, but the same thing. So the fabrication of your material, it's very important to do that to reduce the debye length that you have. And again, of course, the signal that you're generating and how you applying it and measuring it is another factor. So if you're interested when the paper comes, I would send it along your way, and then you can look at it. That was one of the challenges we have, because if it's too wide, it can give you, you can have other ions or coming to you, and you are not measuring exactly the signal that you're supposed to measure, and the noise ratio would be really high.
Well, thank you very much, Professor Hoorfar, and I apologize, Reza and Behdad. I mean, unfortunately, Professor Hoorfar is quite busy. She's the dean.
But please contact me by
Please email her if you
email
have
me
any questions.
Yes, absolutely. And thank you so much, Dr. Salwaji and Dr. Akbari, for this opportunity.
Thank you.
Thank you so much,
again.
Professor Hoorfar. Very interesting talk.
Hopefully one day in person we will have this. Yeah.
For sure. Thank you very much again for your time.
Yes.
And then I'd like to also thank everyone in our audience for participating in this event. And then we'll see you next week.
Take care.
Take care. Bye.